Okay, so in this video, I will go over the steps of how to analyze the antenna angles produced by SwarmSight antenna and proboscis tracking software, and how to basically extract things like the antenna angles as well as density maps and see an interesting response to odors. So what you'll need to do first is we're going to be doing all this in R, and I'll be using the RStudio software. So make sure that you have R installed, and you also have RStudio installed. RStudio is basically a nice GUI for R, and it makes it much easier to work with R. It has a bunch of sort of very helpful features that make it easier to work with. So make sure you have R and RStudio installed. And then afterwards, what I'll do is, in order to, if you want to follow along, what I'll show you is where to download the example code. So you can start by going to swarmsight.org. And once you get there, there's going to be a link called to view on GitHub, which will take you to the GitHub repository for the software. If you've used GitHub before, you can just go ahead and clone it. If you haven't or don't necessarily want to create an account, you can do a clone or download. And once you do that, there's a download zip. So go ahead, download the zip file, and once you've done it, go ahead and extract it. I already did this on my computer, and once you extract it, you should see a folder structure, something like this. So you have the examples, folder, screenshots, setup, and source. Go ahead and open up the examples folder. In there, there's the appendage tracking. And then once you're there, you should see the Jove paper figures folder. And since figures 4 and 5 are probably the most interesting ones and the most challenging ones to create, I will walk you through how to reproduce them. So before I start, there's inside this figures 4 and 5 folder, there's another folder called CSVs, and these are the CSV files of the videos that we have analyzed. So if you just look at one of them, for example, you can see, here's if you watched my previous video, you'll see the same columns that we saw in the last video. And so for each video, we put all these CSV files together, and they're shared here with you on the repository. So so there's a folder full of these files, and then outside of it there's another file, another CSV file called files to run. And if you look at that one, you'll see that has a bunch of columns. So there's a list of these files, which just simply tells this R code to basically look at this file, look at this list, and then process them in this order. And we've labeled them things by like the subjects that we were using, as well as the condition that they were in. And then this column here, almost the last column here, is the buzzer start row. So the buzzer is what we call the LED, and it basically tells you the frame number when for that video when the odor started. So this is important because what we want to do is we want to align all the responses to be aligned with when the odor started. So what we'll be doing here is I've extracted about 300 frames total, 100 frames before the beginning of the odor and 200 ones after. So this number is important to know because it allows us to align those videos correctly based on when the odor started. And for each video, it's going to be slightly different. So for that reason, I created a column, and we actually added all of these the value here manually by looking at each video, so that we know when to start it. And the code that I'll show you will help you assist with that, and I'll walk you through an example how to do that. So before you start, if you have a bunch of video files yourself you've analyzed, you'll probably want to replace this file with the list of your videos, your subjects, and your condition, to make sure that you note which frame the odor started, so that you can align it. Okay, so once you have everything downloaded and you write this folder and you have this R file, go ahead and open this up. it should open up in RStudio. If you don't, you may have to do like a file open type of thing. Once you open it, you should see something like this. So a couple of things to notice. The first one is there's a bunch of libraries or packages that you may need to install if you don't have them already. So in RStudio, there's this packages tab, and you can search it over here to see if you have a particular package. So there's a package TTR. Okay, I have it, but if you don't, you would probably just do install and type in TTR, and it would just download and install that package for you. So you may have to do this for a bunch of these. If you try to run these, I just do Control and hit Return. It basically starts loading those packages, and if everything's okay, it'll sometimes give these warnings, but you can ignore them. But generally, unless they're missing, where you have to download them, it'll be just like this, and you should see no problems with that. Anyway, so let me walk you through the other pieces of it. So the next step is, so what I'll do is I'll basically walk you through this, the code. I won't go too much into detail, but I'll explain a couple of pieces here and there. But otherwise, you basically select the stuff that you want, and then we'll run. So the first thing that I wanted to show you, so we'll just go ahead and run this initialization code. Again, I'm selecting, I press Control and Enter, and that runs the code. You can also press this button after you've selected it. Notice how it reads this files to run file that I just showed earlier. So this is where it will be looking at for those CSV files from each video. Okay, there's another, so this part here, after you select that function and again run it, it will create this function that will help you find that frame where that odor onset goes. So there's a little bit of a documentation here, but basically you can select and then you type in the frame number, or actually the file number. So in that files to run, there's a list of files, and the very last column is the index, which just basically helps you know which file. So this here, I want the 10th file on that list. And if you select this line and run it, you'll see something interesting here. So this here is the buzzer value that you saw over there. So notice how it's kind of steady, and then there's this big bump, and then it goes back down to sort of a stable value. So we know that this is where the buzzer, the LED was on. And the second plot is the derivative of that. So it just basically tells you the change. And what we're looking for is this onset and when it came on, right? When did the odor start? And because we know that in our case we had the odor was delivered for the same amount of time, in each condition we know we don't really care about when it stopped because we know it. But here you can see when it ended. So it ends at around, looks like this is 400. I think this is around frame 500. And if you look over here, it tells you here's the file 10. That was the name of it. This was the file name, and then it changed brightness around 500 and frame 508. So this is around, looks like around 508. So this is probably the correct one. Sometimes you see how there's this other noise in here. It's important to visually confirm to make sure that you are sort of pointing to the correct frame. So what I would do here, and I already did this already, so what I would do is I would go through all these files, 1 through, I believe there's 119 of them. So I'd go through 1 through 119 and do the same procedure and record where this first frame occurs. So in this case, this is 508, so I would write down 508 in that column of that files to run file. So once you've done that and you have that files to run, you can then go on and run the rest of the code. So then this is just a bunch of processing code. You don't really have to know too much about it. You can read through it. I've tried to document this as much as I could so that it makes sense. But you don't really have to know about it. So basically what this will do, it is a big, large for loop which will run through each of those CSV files. I'll extract the frames before and after the onset of the odor, align them all, and basically adjust it for things like rotation, and it will compute the angles. It will make sure the angles are between minus 180 and plus 180. Things like, and it'll plot some of these as well, so you can see what they look like. So what I'll do is I'll go ahead and run all these files here now, and then at the end here, it will pool those files together based on the conditions. So they'll put all the movements, because this is what we're interested in. We want to see is there some sort of aggregate response. So I'm going to pull them together and put them in one place, and we can see what that looks like. So I'll go ahead, select all of this file, all this code here, starting from the beginning till this end data prep part, and go ahead and let it run. And we should see. So yeah, it will show each different plot for each insect and what it looked like. And when it's done, we can go on and look at the pool data, because right now this is just the individual data. So it's still running. It may take a few seconds, but if you want to make this faster, you can turn off the plotting. You may have to comment a couple of lines in there. That way it'll just go faster. Okay, it looks like it's done. And now let's go ahead and make the plots that will generate the angle densities and the means for us. There's this plot quality parameter that I have here. Right now I have it set to 6, but if you make it to 8, it'll make it a slightly higher quality. It'll make much more smoother charts or plots of the densities, but it takes longer to compute. So I will, in this video, I'll just use 6 because it's still reasonable quality, but it doesn't take very long to compute it. So I'll go ahead and run this. This next piece of code here, which basically pulls them together, and then you'll see what it looks like. It creates these 5 plots right afterwards. So I'll go ahead and run it. And okay, it's starting to do it. Okay, so here we have the 5 plots. These are the density plots. One thing to note, and I have a little comment over here, is you should, if you get to this point and you see that there's different scales for this color chart, so we have this situation here, then you should rerun the code again. So notice how we have this is 1 to the minus 6, or 10 to the minus 6, and then this is 1.2 to the minus 6, and this is 1.2. So there's slightly different scales. So it's possible to do this a little better, but it's a bit easier to just rerun it. So there's a little comment that says rerun starting here, so we'll go ahead and rerun this for loop once more. Okay, so I've selected the for loop, and I will rerun it, and our scales should be correct, or the same across all the conditions. Okay, so we have 1.2 to the minus 6 across all of these. Okay, they're all the same. This is important, otherwise it's kind of an unfair comparison. Okay, so in these charts, there's a couple of things. So there's 5 charts because there's 5 conditions. There's the 2 odor, there's the first odor and its diluted version, then the control air, and then there's the second odor, its diluted version, and then the pure version. What we're seeing here, so at this 0 point is basically, or this gray part is where the odor was delivered, was being delivered, and then this is before it, and then this is after it stopped. So these just show us what happens to the antenna movement. So this little black line is the average movement, or average angle, of the antennae of all the insects together. Okay, so you can see it's just kind of noisy here, and then this vertical line is like the average of this of the angles before the onset. And then as we present the odor, you can see that the antenna angle, this is the scale reversed, it increases. So remember, if you watched the first few videos, you noticed that the odor source was coming from directly in front of the insect. So that would be angle 0. If it was antenna were pointing directly in front of the insect towards the odor source, then the angle would be exactly 0. But notice how here the angle goes from around 60 to something like 80. So the angle is increasing. Therefore, the insect seems to be withdrawing their antenna away from the odor source, and then it comes back to the baseline. You can see this also in the diluted version. We don't see the same thing in the air, so we can see that this is not just some sort of artifact of perhaps just air. Again, we see similar things with the heptanal in the second order. So again, it drops down and it recovers, and it looks like the drop down stays a bit longer for the pure version of this order than the diluted version. So you can see that this one kind of goes back earlier. This one takes a bit longer. Okay, so that's just the mean angles. So, and then these density maps, they give us, so for each frame, okay, we can see where the insects tended to have, where the antenna were located. So, I mean, this is very noisy data because the insects are sort of moving the antenna all over the place. But is there some sort of general pattern of whether they prefer one over the other? And if you look at the air, which is the control, you can see that, so the blue spots means there's less, the red spots means more. In the air, we see mostly kind of this middle ground stuff, and you have these blue areas, which is mostly just the artifacts of the density calculation. But for the most part, there's really no, it's kind of evenly distributed throughout the whole experiment. But if you look at heptanal, this second order, you'll see that before the odor is presented, it looks very similar to what you see with the air, which is the control. But then afterwards, after the odor is presented, about 1 second into the odor presentation, you see one is you see that the antenna seems to be withdrawing away from the odor source, and then you see there's a lot of antenna preference for these high angles. So high angles means away, outside, or further down on the sides of the insect head. And this blue little area, it means that there's very few antennae were found close to the odor source. So they all seem to be clustering away from it, and you don't see any antenna across the insects in that condition pointing towards the source. But then this cluster of densities, these density clusters, or in this case like a density hole, you see that it starts to decay, and it becomes back to sort of similar to what you saw before the odor was presented, and then it kind of stays, it kind of recovers, right? So, but interestingly, and so we see the same pattern with the other odor. So here, again, it looks, before the odor is presented, looks kind of normal, but then you see this blue area here and this red area cluster over here after the odor is being presented. And you notice how these clusters seem to be sort of, there's a delay between the onset and when the cluster of these densities appear. So, and in all cases, the blue areas tend to occur about a second or so after the odor is being presented, and similarly with the red cluster. One other interesting thing we noticed is that, for example, in this last odor here, there's this other little area of clusters here, which seem to be related to to how concentrated the odor is. So notice how this was a pure odor, and this cluster seemed to happen later. So it almost like, at first it reaches away from the odor, but then it tries, after the odor is gone, it sort of goes back to the front to kind of check it, perhaps. And notice how the same cluster is present in the other ones, and based on how concentrated it is, it occurs either earlier or later. So that might be it could be a signal that of this sort of rebound cluster might be responding to the concentration of the odor. So basically, this explains the graph that I noticed. So now you know how to do this, how to make the graph yourself. The code is all available online. I showed you where to download it. Again, it's at the beginning of the video. You just go to SwarmSight.org and go to the GitHub repository and download the stuff. And as long as you download the files, look through the comments, and try to follow along to see. And if you don't understand something, feel free to either submit an issue, or if you're adventurous, perhaps even submit a change and a pull request to the GitHub repository, and then we can incorporate your change to this file so that everybody else can see it. Okay, thanks a lot.